Okay, so we have started the drug evaluation topic number 3. So first point we have seen about the microscope morphological or organoleptic characters based on the this one. So how it is evaluated. Second important point is there microscopic evaluation, that is internal based on the internal characteristics. So medicinal plants are there, we are knowing different scientific methods are there. So these scientific methods they are used for the adulteration of the plants. Suppose the stem is there, bark is there, leaf is there, so it is mixed by the other plants are there, and therefore there should be evaluated properly. So which is the original drug and which is the added other drug, other material is there, so which is identical. So therefore internally it is to be tested. So for this one, the medicinal plant parts are there in market, we are knowing some powdered drugs are there. Suppose the clove powder is there, so it will contain the sclerates and the calcium oxalate crystals in the clove, that is lavang. Then Sclerenchyma is absent in the Rauwolfia serpentina root but present in the Rauwolfia micrantha. Generally these species they occur naturally abundant, so therefore they are easily mixed in the powder form. Then secondly, glandular trichomes of the mint म्हणजे पुदिना, and they are the warty trichomes of the senna, म्हणजे senna leaves, सोनामुखी leaves मध्ये warty trichomes असतात, epidermal hairs. Then stone cells, these are the part of the xylem cells, so they are present in frangula bark and absent in the cascara bark. So these are identifying standard characteristics. So standardization of drug is there. Then lignified trichomes and the plasmodesmata present in the Nux vomica seed, it is generally used. So there are the different workers are there, so they have done this one. Then most important part is there, you should remember this. So how internally any drug is studied. So first one is the palisade ratio. Palisade, that is the, it is related with the leaf. So we are knowing mesophyll is divided into palisade and the storage region. Second, stomatal number, how many stomata per square millimeter. Then stomatal index, it is also the related with this one, stomatal condition. Then stomata only, how is the stomata structure. Suppose stomata is shows the, we have seen observing there is the guard cells are there and the subsidiary cells are there, epidermal cells are there. Then Fein islet number, so this practical we are also doing camera lucida. So in square millimeter area how many veins are there, so how it is calculated. So for each drug, leaf drug, generally it is to be important. Then vein termination number, then trichomes or the plant's hairs, calcium oxalate crystals. So these are the microscopic character generally studied for the each drug, right? Then quantitative method, quantitative, that is the quantity. So how it is generally done. So there is the lycopodium spores are there. These lycopodium spores generally used for the standardization. Good example is there, the lycopodium is a fern pteridophyte and these spores are used for the standardization. So one by one we will see about the palisade ratio. So what is palisade? So how many palisade cells are present in the each leaf. For example, these are present below the epidermis in mesophyll upper layer, and therefore the powdered drug, suppose any powdered drug is there, so it is studied under the microscope. So some examples are there, Atropa belladonna, so it shows 5 to 70 palisade cells per fission. Fission means what? In microscope when we are observing, this circle is observing, so this is called as the fission, field of fission. Same thing for the Adathoda vesica adulsa, so 5.5 to 6.5 palisade cells. Cassia angustifolia 5.5 to 10, so this number is there. So it is again for the, so in some leaves upper epidermis generally shows the upper surface shows the palisade and lower surface shows the without palisade but at the lower surface, senna leaves, palisade is present. So these are the good examples. Then stomatal number. So the average number of stomata present per square millimeter, it is called as the of the epidermis, is known as the stomatal number. You remember proper definition, right? Second, there are good example in atropa. Upper epidermis 7 to 10, lower epidermis 77 to 115. Datura metal, datura upper epidermis 147 160. Ossimum म्हणजे तुळस upper epidermis 64 to 72. Lower epidermis 175 to 215. You remember any one example. Then second, so this is for stomatal number. Second, stomatal index. What is index? So it is the percentage proportion of the number of stomata. Percentage in 100. So this total number of stomata. So how it is calculated? So as per with the age of the lip, it is changing. So SI, stomatal index, is equal to S upon E plus S, where SI is the stomatal index, S is the number of stomata per unit area, and the E is equal to number of epidermal cells. Epidermal cells position is outside the subsidiary cells. So good example, upper epidermis stomatal index is the nil, and lower epidermis it is 20.2 to 23. Generally stomata are present at the lower epidermis. Then stomata. Already we have seen stomata character in the epidermal tissue system, so it shows specific character. The main function of stomata is exchange of gases, and therefore as it shows the guard cells, two guard cells always, and varying number subsidiary. So different types of stomata already we are knowing. The moss type, gymnospermous type, gramineaceous type, then dicotyledons. Dicotyledons shows the four types. These are parasitic. Rubiaceae family is the character. Rubiaceous stomata it is called. Good example, senna leaves, sornamuki. Diacytic म्हणजे di means two, so two and the cross subsidiary cells, so therefore this is one type. Peppermint, mentha म्हणजे पुदिना is the good example. Third, anisocytic, that is cruciferous member, मोहरी वगैरे त्याचे example. ते anisocytic म्हणजे there are the two guard cells but three subsidiary cells, one is smaller, one is larger, and one is medium. Then anomocytic, ranunculus type, I think in SYBSc in anatomy we have seen. So please see about their diagrams. So these are the four types, right? Parasitic, diacytic, anisocytic, and anomocytic. Then last one, fifth one is the actinocytic, is additional two guard cells which are surrounded by radiating subsidiary cells. So it is for the stomatal index. Then third, vein islet number. The vein islet number, that is the veins. How many veins are present per square millimeter of the lip surface, either upper or the lower. So generally we are knowing the venation is seen in the lower epidermis. So therefore this is very important to see this character. So good examples are andrographis, paniculata, then bacopa म्हणजे ब्राह्मी, कॅनॅबिस सटायवा म्हणजे गांजा प्लॅन्ट, देन डिजिटॅलिस म्हणजे सोलॅनिसचा मेंबर आहे धतुरासारखा, आणि युकॅलिप्टस म्हणजे निलगिरी. So their number is differing 9 to 12, 6 to 13. So you remember this table. Then vein termination number. This concept is new to you, but see, it is defined as the number of veinlets, terminations per square millimeter, the lip surface between midrib region. Midrib region, from the midrib, how many veins are developed? So this is called as a vein termination. Then one point is also internally studied, calciumoxalate crystals. So in all plants these are not present, but some plants these are seen. So they are having different shapes and the sizes, and therefore the plants are reported. So first one is the cubical, cube-like, as it shows the rhombic diamond shape. Third one, next one tetragonal, onion मध्ये असतात. Then monoclinic, all three axes are unequal in the gall. The gall म्हणजे leaves वरती galls असतात त्यामध्ये. ॲसिक्युलर म्हणजे लॉंग स्लेंडर पॉइंटेड स्क्विल किंवा सिनेमॉनमध्ये असतात दालचिनी. अँड द रोझेट दे आर प्रेझेंट इन द क्लो किंवा अर्जुनच्या लिपमध्ये असतात. So this is the one. It is called as the calcium oxalate crystal. Seventh one is the trichomes or plant hairs. So how are the trichomes? Already we are knowing trichomes are the outgrowths developing from the lip epidermis, upper epidermis. So therefore these trichomes shows the plant hairs multicellular in structure, and depend on the structure they are seen. So covering trichome, glandular trichome, hydathodes, special trichomes are there. I think you should you remembering from SY or FY BSc morphological character. Then in microscopic character, this method is generally studied as the for the histological anatomical character. So the microscope is requiring slide preparation is important. The microscope used for quantitative evaluation all these characters study: stomatal number, stomatal index, phenylate, right? Then stomatal number generally based on the stomata. This is the structure of the stomata showing the stomatal pore opening, then guard cells are there and the subsidiary cells are there, right? Then secondly, there is the stomatal index, total number of stomata per square millimeter. Good examples are the different atropa. Phenylate, how, what is meaning of phen? So just you see, this is the correct mounting in cassia sena 26. So each cell it is to be counted. So therefore this is the one character. Then palisade is present. I have selected this diagram. So upper epidermis, then palisade, chloroplast with present in palisade, and the spongy layer. It is also in total both one is called mesophyll tissue, right? Then in this one microscopy, the quantitative microscopy, that is lycopodium spore method is used. So what is lycopodium? So uniform, all spores in lycopodium are uniform, 25 mm, or average 94,000 spores are present in the 1 mg or 1 gram. So therefore these spores are generally defined as which may be counted single layered, and they are to be used as a standard digestion. So the formula is used for this one. Suppose in ginger powder calculated as follows: N into W into 94,000 into 100 divided by S into M into P equal to percentage of purity of drug. So N means number of characteristic structures, starch grains, etc. in this one. Internally powder drug. For powder drug this method is used. W means weight of the this material. So generally 1 milligram is selected. Then number of lycopodium spores in the same 25 fields. किती फील्डमध्ये मिळाले. नंतर M is equal to weight 1 milligram. So sample calculated on the basis of dried sample, and the P is equal to 2,86,000 in case of the ginger. Starch grains are calculated. So this is about the all about the microscopic characters of the crude drugs. In the next lecture hour we are observing the same topic. The next point is there. Okay, thanks to all, have a nice day.